ticket came in yesterday and said, I've got my genomes, and why can't I build a tree? It doesn't see it. Well, they had submitted their config files and they had them in the workspace. And when they tried to direct the phylogenetic tree to those individual genomes, it wouldn't recognize them. Does anybody know why not? They weren't annotated in the system. Usually every job, every single service in the system, the phylogenetic tree is looking at protein families. It has to be annotated. It has to be translated into BV-BRC for the service to see it. So I had to tell them, you have to annotate them first, and then you can build the tree with them. You can't just have contigs out there. It's not going to work. This is one of the few places that you can bring in outside data and compare it to data within the resource. And I say that, but I'm going to direct you to a file that I have in the workspace. So, and we're going to navigate to, so click on the folder icon, and then we need to click up here because it's in the public workspace. So click on the down arrow up at the top here, and then click on public workspaces, yes? And it takes a while for it to figure it out. Ah, good. And then scroll down. And you know, we didn't talk about this much yesterday. What we're seeing here is, well, Malik talked about how you can make a public workspace by clicking on that box that everybody can see. And I have a class on Coursera, free class, Bacterial Bioinformatics. And one of the assignments is that you create a public workspace. And I can tell that these people have been very busy right there. It has that icon. The global icon means it's a public workspace that everybody can see. If there's an icon that looks like a little human with people around it, that means it's a shared workspace that's shared with you, so you can see it. And then your private workspace only you can see. So click on BV-BRC Workshop, click on Proteome Comparison, click on Ricea, and click on this Ricea Unique Protein Families FASTA, and then click OK. Hello, where are you, my friend? One thing we could do is download it and then upload it, but also note while I'm here and we're trying to get through this problem is that you could also add these things here to this, because this one has 7 genomes. It can go up to 9 compared to the reference. So I could add other things here too if I wanted to. I could add a feature group. I could add a FASTA file to see that as well. One thing I didn't point out that I want to point out is if you click up here under the advanced parameters option, you can play with the BLAST analysis and try to see things that are more specific or less specific. So you can dial up the minimum coverage, the minimum identity, and the E-value if you want for these things. I use this tool a lot. I told you guys yesterday, I do a lot of work with Brucella and with the lipopolysaccharide code, and I was talking to you about how I use the protein family sorter for that. One of the things that they use to distinguish Brucella are these lipopolysaccharide genes. So I have the feature groups of those, and I often use this tool as my first pass to see with a new genome, does it have the genes and how strong are they? And that's the kind of thing with Brucella that you can build a publication around. Okay, but for those of you who can submit that job, go ahead and do that. And we'll look to see if any of the previous jobs submitted. And if not, there seems to be a bug that's preventing this, I imagine, and they're trying to figure out what it is. So some of you were lucky and the bug didn't attack you, and some of you were like me, unlucky. So you can click on the jobs. Mine have already completed. That's another good thing about this service is it's really, really lightning fast. Well, okay, it's not lightning fast, but it's pretty fast. How many people have had one of those jobs complete? How many people have not had a single job complete yet? Okay, so the all genomes job should have completed for all of us. So let's highlight that row and click on the view icon. And this tells you all the files in the downloads that come with the job. And you can read the tutorial to see what all of those things are if you want. But the most important thing to me is always up here, these two for this particular. And I recognize that there are some issues, right? Sometimes we ask you to click on the view icon up here. Sometimes it's in the vertical green bar, but when you see something up here, it's usually say there's a special treat behind it or something that you want to see. So let's click on this blue view button here. And there it is. This is the proteome comparison visualization. Now across the top, the colors are indicative of the strength of the hit.
We have a question here.
Yes, there's a question from the audience.
Forgive me, I've never been exposed to this type of analysis. Why were those 7 genes chosen?
Just those 7 genes, there was really no reason I chose those genes. You'll be able to see, because I wanted to show you what the visualization looked like. Those 7 genes really aren't of interest to me. The only interest they had was that I knew that there would be hits. So I wanted to show what the visualization looked like for this particular example. However, when I'm doing things like with brucella, I use those LPS genes, and those have tremendous impact for me and for other people who look at brucella. Those are the first thing everybody looks at. So that's why I chose that. This is just to show you a picture of what it could be like with your data.
So if you were studying a model organism in your lab or an organism, a pathogen or organism of interest, you would just curate a list based on what you know about the biology of the organism. Which
or if they were virulence factors that are really important, like maybe toxin genes or something that you really care about and you want to see, first of all, if they're there, but also this will tell you there's an indication of it here in the strength of the blast hit. But it can also give you a sense when we dig into the Excel file that comes with this, it'll show you sequence, identity, and coverage so that you get a sense where these genes are falling apart too and things. And that, it sounds ridiculous. Well, it doesn't sound ridiculous, but I can remember working with this one guy who works with Mycobacterium tuberculosis. And there was one gene, he was on the phone with me and he said, could you just look at this gene that I care about? He had asked me to annotate one of his organisms that was much more slow growing than the other ones. And he couldn't figure out why. And he just said, how about this gene? And I looked at it and I said, oh, look at that. It's truncated, it's short. And then I went into the conserved domain database, which from the feature groups, we have a direct link to from any feature page. And I said, oh, look at that. This main domain that's at the front of the gene, it's been truncated, so it can't do that. And so that opened up everything to him. And that was a paper in PNAS. And I got on it just from this phone call, from sitting there and talking to him and looking at this thing. So that can be the power of this kind of analysis.
So in that analysis, he had a gene, there was a hypothesis that a particular gene was involved in altering the growth kinetics of the organism. So he compared it to the reference genome with his isolate to the reference genome to see what the difference was.
Well, when he asked me to look at it, because, like this is like an encyclopedia of genes when it comes back with a genome. And as somebody who doesn't look at those, I'm not an MTB expert. I could have looked through each of them individually, but he had great knowledge. And this was a gene that was of interest to him. And so he's talking to me about this and he says, can you look at this particular gene? He's a lab guy. He doesn't do bioinformatics or anything. So I said, sure, no problem. So I looked at it, and then me looking at it, he wasn't looking at it and saying, my gosh, look at the strength of that blast hit and that sequence identity. I was driving. he was in the back seat and I was saying, oh, look at this and this. And then to him, it was like, ah,
So I understand that particular use case. So in that instance, he was comparing the genome of an organism that had altered growth kinetics
right.
to the
exactly.
reference genome with respect to this one
Right.
gene of interest.
Right.
Thank
He
you.
knew both of those. This originally started with the participant in the audience asking me, why are these genes I have had you all select interesting? Maybe to somebody, not particularly to me, because I'm not working on a publication here. The main reason for me to choose these was I wanted to show you the visualization that's possible for these. And I had to choose something that some of them have, or it would look dumb. So I wanted it not to look dumb for you. So across the top, it's showing you the strength of the hit. Blue is strong, red is very weak. It's showing you the tracks. So actually in the genome group, I had BRHL. So I used it as the reference. Then it's to itself. So naturally, it's going to be extremely strong. The bidirectional blast hits are going to look great when it's looking at itself. But then this is the order. Here's my gorilla strain. Here's a human louse strain. Here is a chimpanzee strain. Here's a human louse strain. Here's a chimpanzee strain. And then here's a human louse strain. Now notice I had you guys do the genome group, right? So remember, we're moving toward publication here, supposedly. Well, we're not, but when it's your stuff. When you add the whole genome group, it may not give you the order you want. However, when you resubmit the job, when you're thinking publication, you can go in and add them one at a time and then get them in the right order so that it looks pretty. So I would do like human, 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 chimpanzee, chimpanzee, gorilla, gorilla. So then you'd see blue fading to green and then finally down to the red, and it would be lovely. But this one is not showing you that. But you can see things too. You can see where it's missing large sections in the gorilla strain that the other ones don't have. And you can download this as an SVG, and you can see it's got a single contig, so it looks lovely. But that's just a visualization, right? It's just a picture. If you mouse over these things, you could jump to that particular location. If I was interested, I could click on that. It's going to take me to the feature page for that individual gene. Let's go back because that opened a new tab. We can go back to the previous tab. So this is just a picture, but we also provide you with all the data underneath that. So let's go into the downloads and let's click on the genome comparison table. We have a question. Yes.
In this plot, what is each bar?
Is a single gene.
The gene, and the thickness represents how long?
No, the thickness has, because this is a genome with thousands of genes, right? Each of those, the thickness indicates that there are a bunch of genes close together. Each gene will be small, and we'll be able to see size in some of the other examples. We'll get there in a second. But right now, it's just showing you the location on the genome. And when you see something that looks really big, like, is this really big? This is a collection of mini genes. So if I'm careful in my scrolling, you'll see that there are a bunch of them there. Does that answer the question?
Yeah, this was like a protein analysis, but it's...
Yes, it's a bidirectional protein blast. So it's taking the reference genome and the comparison genome, and it's blasting against it. And so what you'll see is, here's the reference and here's the comparison. And the reference genome may say, oh, this gene is my best match in the entire genome in this comparison, but it's bidirectional. This comparison gene may say, no, I like this guy better. So you'll be able to indicate where you might have paralogs, homologs, or there was a duplication event and maybe somebody lost one and it might be the best hit here, but this guy may say, no, this is my best hit and you're the duplicate. Does that make sense?
That makes sense. My last follow-up question is it looking at express proteins or is it doing this
No,
like?
nope, it's all genomes. It's not expressed. And it's not looking at the strength of expression or anything like that. To do that, you would need the RNA-seq tool, which Clark will be doing at the end of today.
So it's just scanning through the genome.
Right, and looking through things. So let's click the download icon and click on Genome Comparison Table XLS. And that's in Excel. We also provide it in text if you want it that way, which I never do. Okay, show in Finder. There we go. So this is a very complicated file, but I'm going to convince you that it's one of the most beautiful documents that we produce. And you might want to throw out every book in your house and then just print this up and have it for your perusal. The very top row is showing you the genomes and the analysis. And the first one is our reference genome. Let's go down to the second row. And the first one is telling you the contig that it's on. If this is in multiple contigs, this is important so that you know where things are. Okay. The second column is showing you the gene number. It's starting at the first and working its way up in the reference, and it's searching for its best match. This is like a gene dating service, right? So it's like eHarmony or something. It's trying to make the perfect match for these things. This one is showing you how big that gene is. Why would you want to know that? When you're looking at the comparison gene, you might see that one's really small and one's really big, and that might be an indication that your gene is truncated, something like that. The next one is the unique identifier. If you go in and click on any of those, it's going to open a tab because those are all hyperlinks for that particular gene in that reference genome. If that genome has a ref-seq locus tag, because we recognize that those are gold to the research community. Everybody talks in ref-seq and GenBank locus tags. So we don't forget those. We have those here so that you can refer to them. If it has a gene name or a gene symbol, it provides that. If it had a local protein family ID, it provides that. If it had the global protein family ID, it provides that. And you can use these protein families like in the protein family sorter that I told you guys about yesterday. Those can be valuable for you. The name of the gene, where it starts, where it ends, what strand it's on. Now we're moving into the comparison genome. And it starts, it's saying, how good is this hit? And is it bidirectional or unidirectional? And not only that, you can notice in the first one down, it's unidirectional and it's telling you that it's going this way, that the reference genome, it's a stronger one in that. And then it's showing you the contig on this second genome, the gene number, the amino acid length, the gene ID, the locus tag, the gene name. But now this is where the meat of it is, okay? It's showing you the percent identity and the sequence coverage. So it's showing you the strength of the blast hit. It's all here in this. And when we get to this next one, this is the gorilla genome that was missing a lot of stuff, but it repeats itself. So one of the things I'm going to do that I often do is I'm just scrolling over all of these, and then I'm going to shrink them up really small. And then I have sort of like a bird's eye view of these comparisons, and I can quickly scroll through these genomes and see who has what and the strength of those hits. And things like this, I mean, it's not in this case because these guys are endosymbiont, but often where you see these big blank regions, sometimes those are indicative of areas of lateral transfer, horizontal transfer. When I see something like that, I go in and I look and see if there's any tRNA genes in the flanking areas, because often those things are jumping into genomes in those places. And I just learned to do this from Brucella, because those lipopolysaccharide genes that I talked to you about earlier, those are associated with tRNA genes, and they hypothesized they came into Brucella by lateral transfer. So it's just something I'm inclined to do. But then you can quickly see who has what. And then you could see flanking regions too, for areas where it's strongly conserved in all of them, and then it breaks off in others. Does that make sense? Yes, question. How did I do that? See, they're all selected here. And when they're selected like this, I can go over the last one and I can change the size of it and I can shrink it up. See, it's those kind of skills that I have that have people thinking that I'm a computer scientist sometimes with the Excel sheet, which I'm not. I just know how to manipulate Excel. Okay, so this is what it looked like when we're looking at the genome versus genome. So let's go into Workspaces, BV-BRC Workshop, Proteome Comparison, Ricea, and then let's look at the one we couldn't see, genes of interest, this checkered flag, the second one. And click on that, and then let's just click on the view icon. Okay, so this is those 7 genes of interest, and when you're using a feature group, the feature group has information about that gene. So when you're looking at that, the service is trying to place where those genes are in the genome. And so it's resolving that. So what you're seeing is the reference genome for those 20 genes, and here's where they are. And then as you march your way through, it's the order of those, the 7 genes, and then we have the gorilla strain, and then we have a chimpanzee strain, and then we have a human strain. So anyhow, it's stepping down that. So I wanted you to see what it looks like when you use a feature group. And it's different, right? But this might be something you might want. Sometimes it's interesting. This is an interesting way to look at. Imagine if you're interested in a pathway, and often in bacteria, but not always, they're trying to assemble the genes that they're using together all on the same place on the contig, so that, it can just run right through, or maybe looking at an operon or something like that. So you might want to show it that way. Or it might be disparate. So that might be a reason why you might want to look at these things. If I were looking at the Brucella LPS genes, most of them would be right here. And then there would be 2 over here and a couple over here, because they cluster them all together. They came in by lateral transfer and they're all together in the same place. Let's go back up to Ricea. So click on this one, this breadcrumb, and then click on the last flag, unique protein family, 7 genomes. This is the one I couldn't get, but it was the FASTA file. I mean, I obviously got it at some point, but it was buggy today. And click on that. Those are the same genes. But now this one, it doesn't know, because they're FASTA files. It can't place them on a genome. All it's got is sequence. And so this time it's showing you the size of those. So this is a very big gene. This is a very small gene. I just wanted to let you guys see how you can game this thing to get a visualization that you're interested in, that you might want to include in a paper. And it shows you the data all behind that.